Imagine getting your medicine, maybe a vaccine, maybe something you need long term, not with a shot, not with a pill you have to remember every day, but with a simple patch, like a Band-Aid almost.
Yeah, just stick it on and you're done.
Exactly. And for years, that's been the promise of these things called transdermal microneedle patches. Tiny, tiny needles you see, so small they apparently don't hurt, delivering medicine right through your skin.
It sounds pretty revolutionary, doesn't it? The potential to change how we deliver everything from vaccines to complex therapies is just enormous.
Absolutely immense. But despite all that excitement, actually getting these patches into widespread use, well, it's hit some major snags, some really tough roadblocks.
That's right. I mean, how do you actually cram enough medicine into something so small?
Mm.
That's been a big one.
And how do you keep sensitive drugs, like a lot of vaccines, stable for months without needing a fridge
The
constantly?
cold chain problem, yeah.
Yeah.
Huge logistical challenge.
And maybe the biggest one, how do you make one single patch application last so you don't have to keep putting on new ones all the time?
Right, avoiding that frequent redosing.
So these issues have kind of kept microneedle patches more in the realm of like cool concept rather than everyday reality.
Until maybe now.
Exactly. So our mission for this deep dive is to really explore how some, frankly, groundbreaking research is finally smashing through those barriers.
We're going to focus particularly on the work coming out of Thun Nguyen's lab. He's at the University of Connecticut.
Yeah, we'll unpack the innovations from his team and see how they're turning these microneedle patches from, let's say, a distant dream into something that could be a powerful, practical tool for medicine. Pretty soon, maybe.
Sounds great. Where should we start?
Okay, so let's start at the beginning. Why are these patches such a big deal in the first place? What are the core advantages over, say, a regular needle? And then we'll dig into those limitations you mentioned.
Well, the elegance is really something. They're designed to be minimally invasive. The needles are so tiny, they only pierce the very outer layer of skin, the stratum corneum, and they don't really hit the nerves underneath.
So genuinely painless, or close to it.
Pretty much, yeah. Research, like some published in Molecular Pharmaceutics, really highlights this. And because it goes through the skin, you can get a much steadier level of the drug in your system over time.
Okay, steadier levels, that makes sense. What else?
A really key thing is they bypass what's called first-pass metabolism. When you swallow a pill, it goes through your gut and liver, and the liver can actually break down a lot of the drug before it even gets into your bloodstream.
Ah, so you lose a chunk of the dose right away.
Exactly. With a patch delivering through the skin, the drug goes more directly into circulation. So that's a big efficiency gain for certain drugs.
Makes sense.
And then there are the practical things. You could potentially apply it yourself, right? Less need for clinics or nurses for simple applications.
Which saves time and money.
Definitely. And another point often mentioned, no sharp needles to dispose of. No biohazard waste in the same way as traditional injections. That's a not insignificant benefit for safety in the environment.
Okay, so painless, steady dose, avoids the liver issue, self-administered, less waste. Sounds like a slam dunk. So what were those big hurdles holding it back?
Well, despite all those upsides, getting them widely instead ran into some serious walls. The first one, as we hinted, was often really low drug loading.
Just couldn't get enough medicine in there.
Right. The common manufacturing methods, like using liquids and casting them into molds, just physically limited how much drug you could dissolve and fit into those tiny needles.
Yeah.
We're talking less than a milligram, often in a standard 1 by 1 centimeter patch.
And that's just not enough for a lot of things.
Not nearly enough for things like antibody therapies or even some vaccines that require a bigger initial dose. You'd need a massive patch or multiple patches, which starts to defeat the purpose.
Okay, so low dose capacity. What was next?
The other huge one was thermal stability. Think about most vaccines or other protein-based drugs like antibodies. They're delicate molecules.
Right. They need to be kept cold, the whole cold chain logistics thing.
Precisely. They need strict, often refrigerated or frozen, storage to stay effective. But the problem was, some of the manufacturing processes for microneedle patches themselves could involve heat, sometimes up to 60, even 80 degrees Celsius.
Oh, wow. So you destroy the drug while you're trying to make the patch?
Potentially, yes.
Yeah.
Those temperatures can easily damage or inactivate these heat-sensitive proteins. So making a patch that could actually sit on a shelf at room temperature or survive shipping without refrigeration, that was a massive challenge.
Okay, dose loading, stability. You mentioned a third one.
Yeah, the release profile. Most of the earlier microneedle patches were designed for basically immediate release. The drug dissolves quickly once the needles are in the skin.
So like a quick shot, but through a patch?
Kinda, yeah. Which is fine for some things, but if you need a drug to work over days or weeks, you'd still be reapplying patches frequently.
Which, again, sort of undermines the convenience factor. If you're swapping patches daily, it's not quite the revolution promised.
Exactly. So you had these three core problems: getting enough drug in, keeping that drug stable without a fridge, and making it last from a single application. Pretty fundamental obstacles.
They really sound like it. So how on earth did Dr. Nguyen's team at UConn start to unravel these? I mean, tackling all three, that's ambitious. Where did they begin?
It really is impressive work. Thanh Nguyen, he's an associate professor there in mechanical and biomedical engineering, and his group really systematically went after these core issues. Let's start with that first one, getting more drug into the patch, the ultra-high drug loading.
Okay, how do they do it?
They essentially threw out the traditional liquid casting method, or at least reimagined it fundamentally. Instead of dissolving the drug in a liquid, they developed something called a powder filling technique. Yeah. As they described in their paper in Molecular Pharmaceutics, they literally take the drug in its dry powder form. They used human immunoglobulins, or Haig, as a model, and they fill it directly into the tiny microneedle molds using a vacuum.
So packing the dry stuff right in instead of trying to dissolve it first.
Exactly. And just switching to this mechanical packing method was, well, a game changer.
How much difference did it make?
According to their results, it increased the loading capacity by about 20 times. 20, compared to the old solution casting methods.
20 times. That's huge.
It really is. They managed to pack around 4.5 milligrams of this model antibody, HIGG, into just a 1 by 1 centimeter patch.
Which is getting into a much more useful dosage range.
Definitely. And they also think about scalability. They describe these 1 by 1 centimeter patches almost like pixels or building blocks.
Oh, like Legos?
Kind of like Legos, yeah. You could assemble them into larger arrays. So a 2 by 2 centimeter patch using 4 of these pixels could potentially deliver hundreds of milligrams of an antibody.
Okay, now you're talking about doses relevant for things like cancer therapies or maybe HIV prevention antibodies, right?
Exactly. Those often require really high doses, and this powder filling approach opens the door to delivering them via a patch.
That is genuinely astounding, that 20-fold increase. Is that something they think can be reliably scaled up for manufacturing? And what about keeping that powder stable? You solved the loading, but did the powder itself hold up, especially outside a fridge?
Great questions. On the scalability, the techniques they're using, like vacuum filling, are actually pretty established manufacturing processes, just applied differently. So there's optimism there. But your second point hits the nail on the head: stability. That was the next mountain they climbed.
The cold chain problem.
The cold chain problem, exactly. Because even if you can load the powder, if it degrades at room temperature, you haven't solved the real-world logistics issue. We know traditional vaccines, most protein drugs, need that constant refrigeration. It's a massive cost and logistical headache, especially for global health initiatives.
So how did Nguyen's lab tackle that?
They focused on finding the right excipients. These are basically inactive ingredients you add to stabilize the main drug.
Like stabilizers you find in food sometimes?
Sort of, yeah. They screened various options and landed on a specific combination: trehalose and sucrose. Just simple sugars, basically. Let's call it TS.
Trehalose and sucrose. Okay. What did TS do?
What it did was pretty remarkable. Their results, again in Molecular Pharmaceutics and also highlighted by UConn today, showed that formulations using this TS combination successfully stabilized almost 100% of the human immunoglobulins they tested.
Okay.
At temperatures up to 100 degrees Celsius for at least an hour.
Wait, 100 degrees Celsius. Boiling point.
Boiling point for an hour. And perhaps even more relevant for practical use, the formulation kept the antibodies stable at body temperature, 37 Celsius, for at least 3 months.
3 months at body temp. That's incredible. That completely bypasses the need for refrigeration during shipping and storage, doesn't it?
That's the potential, yes. Imagine mailing vaccines like regular packages. No more complex cold boxes, no more worries about power outages breaking the cold chain. It could revolutionize vaccine distribution, especially getting them to remote or resource-limited areas.
Well, absolutely revolutionary. Did they test it with a real-world type of molecule?
They did. They used an antibody called B12, which is known to neutralize HIV-1, the AIDS virus. They loaded this antibody into their microneedle patch using the TS stabilizers, exposed it to heat 37 degrees for a month, and then tested its activity in vitro in the lab.
Yeah.
It still worked. It maintained its antiviral activity. That's a strong proof of concept that this stabilization technique really protects the function of these complex therapeutic proteins, even when loaded into the patch and stored warm.
Wow. I mean, the implications are just staggering. Anyone who's dealt with the logistics of vaccines or even just needs to keep their own insulin cold immediately sees how huge this is. It changes the whole game.
It really does.
Okay, but you mentioned that third hurdle earlier. Even if you load it high and keep it stable, most patches just dissolve quickly. How do they get around the need for frequent reapplication? How do you get long-lasting effects from one patch?
Right. That's the final piece of this elegant puzzle. And this might be the most ingenious part for patient convenience. They developed what they call a core shell microarray system.
Core shell sounds like a candy.
Huh, maybe a bit. But the idea is that each individual microneedle isn't just a solid piece of drug. Instead, it has an inner core made of that dry, stabilized drug powder.
Okay, the stuff we just talked about.
Exactly. But then it's encased, or coated, in an outer shell made of a biodegradable polymer. Specifically, they use PLGA, which is a well-known material already used in dissolvable stitches and controlled-release drugs. It's known to be safe and break down predictably in the body over time.
So the shell dissolves slowly.
Precisely. And the really clever part is that they can tune the properties of that PLGA shell, its thickness, its composition, to control exactly when it dissolves and releases the drug core.
Ah, so you could design different needles on the same patch to release at different times.
That's exactly what they did. They ran studies in rats, which are obviously a crucial step before humans. They showed they could get controlled, dose-dependent release of the human egg antibody.
Okay, controlled release is good, but did they achieve that long-term effect?
Yes. This is the really impressive part. They designed 3 different sets of patches within a single application system. One set released the drug immediately, like a priming dose, on day 0. Another set was designed to release its payload around day 10. And a third set released around day 21.
Like timed bursts of medication from one patch.
Exactly. Multiple injections simulated by one single patch application.
And the result?
They were able to maintain the antibody levels in the rat's blood at a consistent therapeutic level for a full 25 days.
25 days from one patch application.
25 days from one patch. Just stick it on once.
That just blows away the need for booster shots or frequent clinic visits for so many things.
That's the goal.
Yeah.
As UConn today pointed out, this could be huge for compliance, especially in places where getting back to a clinic is difficult. It really moves towards a set-it-and-forget-it model for therapies that need sustained levels.
Okay, wow. So let's try and pull all this together. We've got the powder filling for ultra-high drug doses, 20 times more. We've got the trihalose and sucrose for amazing heat stability, no more cold chain. And now this core shell system for single application, long-acting, multi-burst delivery.
A trifecta of solutions, really.
It really is. So what does this actually mean for us, for you listening? What's the bigger picture for healthcare, for fighting diseases globally?
Well, if you connect all these dots, the work by Fan Nguyen and his team at UConn. And it's worth noting they've had support from groups like the USDA and the Bill and Melinda Gates Foundation, who are obviously very interested in these kinds of practical solutions. It fundamentally tackles some of the biggest practical headaches in delivering medicines today.
It sounds like it breaks down major barriers to access.
Hugely. Think about it. Self-administration. No need for refrigeration. Only needing one application for weeks or maybe longer. That dramatically improves who can get vaccines and therapies, especially in remote areas or during emergencies.
That idea of mailing vaccines like a simple bandage, it keeps coming back. It's such a powerful image.
It really is. And beyond access, think about patient compliance and the burden on healthcare systems. Fewer clinic visits, less discomfort from needles, easier logistics. It all adds up to making healthcare more efficient and, frankly, more patient-friendly.
And the potential impact on diseases.
Immense. The idea of a single patch providing long-lasting immunity could be transformative for efforts to eradicate diseases like polio, measles, rubella, maybe even future pandemics like COVID-19.
And it's not just vaccines, right? You mentioned antibodies.
Absolutely. Their papers make it clear the potential applications go way beyond vaccines. Think about treating or preventing HIV, certain types of cancer immunotherapy that require high antibody doses, managing autoimmune disorders. Anywhere you need to deliver a large amount of protein therapeutic, potentially over a long period, this technology could be relevant.
And crucially, is it safe, comfortable? People won't use it if it irritates their skin badly.
That's a fair point. The studies they published did look at skin irritation in the animal models. They reported only minimal and transient redness, basically slight irritation that went away quickly. That suggests good potential for patient comfort and acceptance. It has to be user-friendly to be truly successful.
It really does make you stop and think about how different receiving medication could be in the, well, maybe not so distant future.
It's a paradigm shift, potentially.
So just to recap the key takeaways here, Sand and Nguyen's lab at the University of Connecticut seems to have really cracked several major codes for microneedle patches. There's that ingenious powder loading technique for getting huge doses in.
20 times the load, yeah.
Then the power of simple sugars, trehalose and sucrose, to make these delicate drugs incredibly heat-stable, ditching the cold chain.
Stable for months at body temperature.
And finally, that clever core shell design allowing for multiple timed bursts of medicine delivery over weeks, all from just one single patch application.
Putting it all together for a truly practical system.
Absolutely. So think about this. How could this bandage-like future of medicine, this tech that seems almost like science fiction, not only make our own lives simpler, but fundamentally shift the balance in global health, making truly vital treatments accessible to literally everyone, everywhere? And maybe, just maybe, what other really complex medical challenges out there could be transformed if we keep rethinking not just the drugs themselves, but how we actually get them to where they need to go inside our bodies.
That's the exciting frontier, isn't it?
Yeah.
Delivery methods can be just as revolutionary as the medicines themselves.